Today we're going to be discussing the neural mechanisms behind emotion. We're going to be mostly focusing on the amygdala and its impact on how we perceive emotions, with a lot of the focus on fear. So why don't we get into it? So what is emotion? Well, it's a complex physiological state that involves three distinct components. Subjective experience, such as feelings, like if you feel angry, if you feel sad, if you feel disgusted or surprised. We can describe these things, and they're subjective. They're with an internal state, a description of our internal state. There's a physiological response that goes along with emotions too. Our overall physiology will be different if we are angry or sad or happy or surprised. And then behavioral or expressive response is another core component. We're looking at these faces here. We have three individuals. They have obviously distinct features, but we can look at how they are expressing themselves, and we can infer very accurately what emotion they're actually expressing. And this is a behavioral response. The way we contort our face communicates to the people around us what emotion we are feeling. What are our subjective feelings, and tells them something about our physiological response so that they can respond in kind. So there's several basic types of emotions that are recognized, and this is across human cultures. Fear, disgust, anger, surprise, happiness, and sadness. This is all the study of emotions falls under the term affective neuroscience. So that's the study of the mechanisms of emotion. Affective comes from the word not effective, but affective. It comes from the term affect, which is the experience of feeling, or the experience of emotion. That's referred to as affect. We will be talking about affective disorders when we talk about mental disorders, but this is another important aspect of this, and they refer to emotional disorders such as mood disorders. Examples are major depression and bipolar disorder and anxiety disorders. So these three are things that can have a significant impact on our affect, and we will talk more about this after Thanksgiving. Getting into emotion a little bit more, that it has this it's this cross section between behavior, the thing that we can see, the underlying physiology, and feeling. And it's this complex physiological state involving these three distinct components. So a little bit more about the subjective experience is what emotions you feel and to what degree. Is an angry person merely annoyed or enraged? Emotions can also be multidimensional, involving multiple emotions simultaneously, right? You can be angry and disgusted at the same time. The physiological response is also core to this. For instance, maybe your stomach is clenching. Maybe you're having heart palpitations, sweating palms, rapid breathing. All of these related to the sympathetic nervous system. You can have other changes that are outside of the sympathetic nervous system, but are still physiological changes. And then the behavioral or expressive response, this is the verbal. You can have verbal component to this. Someone can express that they're angry using their words, or express that they're happy with laughing. And then there's nonverbal components to this, of course, as well, such as your facial movements, smiling, scowling, and behaviors like crying, laughing. These are core behaviors that are cross-cultural and will tell the people around us, family members, friends, exactly how we're feeling. So there are two dimensions in which a particular emotion can be broken down. one of which is valence. And so we can categorize the experiences and feelings and emotions of one person having being either positive or negative along this dimension called valence. And the other one is arousal, so the degree to which it has an effect on our overall physiology. So certain emotions can have relatively low impacts on our arousal and have a lower impact on our primordial parts of our brain, or they can have a high impact and can activate the more physiologically relevant portions of our brain, such as instigating survival instincts like fight or flight, or inducing, say, extreme euphoria, such as laughing out of control at a really funny joke, something like that. And so here are a few examples of how this can follow along, right? So we've got valence here and chair. So I assume this means like just sitting in a chair is not something that's going to be positive or negative. I guess if you've been standing all day, sitting in a chair might be a positive experience. But whatever. So we're going to say sitting in a chair is largely not positive or negative, and it doesn't really have much effect on our overall arousal state. So that's sort of the control circumstance here. But then, so we can talk about other experiences and the impact that it'll have on the valence dimension and arousal dimension, such as seeing a beautiful sunset. This is something that for most of us isn't particularly arousing, but you know, it's a positive experience and usually brings about happiness. Seeing a smiling baby can be even more arousing, obviously depending upon the circumstances, but this is usually an exciting thing, especially if the baby's smiling for the first time. So it'll be very arousing and very positive. And that will be, you know, that's usually a very joyful experience for people. Then, you know, there's a category, several categories of basic emotions which we can subdivide that generally are negative: sadness, fear, anger, disgust. So these will have a negative valence. But they can vary in their degree of arousal as well, obviously. So funerals are a particularly sad experience. The behaviors associated with that will be related to sadness, such as crying and sobbing and showing sad facial expressions. But they're not particularly arousing, right? They're not going to be inducing fight or flight experiences. But seeing something like a snake is a very fearful experience for many of us. It's not terribly negative, but it's on the negative side, but it can be a very fearful experience. And then eating something that is rotten, that will elicit the emotion disgust, and obviously is a very negative experience. I mean, I would put that below. I mean, yeah, it's on par with funeral. Funerals are terrible too, and but that's a particularly arousing experience too, right? Like vomiting. People react very strongly when they eat rotten food. So you can think of any kind of experience that one has could be plotted on this two-dimensional scale. So this is a simplification, but it does tend to fall into lines with the negative emotions, negative experiences leading to certain categories of experience that are considered to be sort of negative, and then positive experiences leading to the kinds of emotions that are positive. Now, one key part of understanding emotions from the neurobehavioral perspective is looking at behavioral responses. So it's one thing to inquire about one's feelings, and it gives us important information, but we can observe in great detail and even quantify the overall behavioral response by looking at someone's facial expression and the other behaviors that they elicit. So a critical nonverbal expression of emotion is through facial expressions, and there appears to be specific brain areas involved in relaying facial expressions that are involuntary. One way that this is obvious is looking at the smile. You have all seen this. You're trying to take a picture for Instagram or whatever, the snap book or whatever you kids do, and you know, you got a big group of friends, but you're like, okay, you gotta give me a real smile. This is hard to do for a lot of people because there's a difference between a genuine smile and a voluntary smile. And you can look at we can look at a face and know whether someone is giving us a genuine smile or a voluntary smile. We can look at these faces here, here, here. And it should be pretty obvious. This is the genuine smile column, and this is the voluntary smile. And when you ask someone, hey, okay, smile, give me a picture, this is the first thing that they're going to do, usually. This is the voluntary smile. But a genuine smile looks different, and you can just look at the different facial features to see this. Some of it has to do with the eyes. Some of it has to do with the way the mouth is pulled up. There's a difference. And it turns out that there are different brain circuits that help regulate the smile, even including just in the mouth. So we're going to talk about that in the next slide. In this slide, we're going to look at the smiles of two different patients that are exhibiting a form of paresis. Paresis is the medical term related to, means muscle weakness. And so this is facial motor paresis, so this is weakness of the face. So this is the face of a patient who had a lesion that destroyed the descending fibers from the right motor cortex, displaying voluntary facial paresis. And so when this person was asked to show her teeth, the patient was unable to contract the muscles of her left side of her mouth, right? Right motor cortex, opposite side of the face. So she could not show the left side of her mouth. She was asked to give a voluntary smile and she couldn't do it. Can't see the teeth there. Yet, she could give a spontaneous smile in response to a humorous remark that was nearly symmetrical. So it's not perfect, right? There's some overlap here, but you can see that this smile is basically pretty good, whereas this, the voluntary smile, is severely distorted. Despite what superficially seems like trying to make the same basic movement, it's actually quite different. So her voluntary control was affected, and since we know that voluntary control is regulated by the motor cortex, it makes sense that the voluntary smile is only showing half, given that there's damage on only one side of the brain. Yet the emotional smile that is more instinctual and spontaneous is mostly symmetrical. So now we're going to look at the smile of someone who has emotional motor paresis. So this is the face of a child with a lesion on the left forebrain that interrupted descending pathways from non-classical motor cortical areas. So this would be other areas outside of traditional motor cortex where voluntary motor control is regulated. This boy had emotional facial paresis. So when the child was asked to smile voluntarily, the contractions of the facial muscles were nearly symmetrical. You could see, like, smile for me, looks perfectly fine. In a spontaneous response to a humorous comment, however, the right side of the child's face failed to express emotion. So you can see, because this individual had damage on the left side of the brain, it was his right side of the face that was affected, and he cannot create an emotional response on the left side of his face. So it's separate neural circuits as the origination points for these two different pathways. This is a clear demonstration that there must be separate circuits that are involved with this. So voluntary movement coming out of the motor cortex, going through the brainstem, this is voluntary facial control. When there's damage in this pathway, this leads to voluntary facial paresis. This is referred to as a pyramidal smile because the circuits that regulate this go through the medullary pyramids. And so it's going to affect the motor pool in the facial nucleus. So, because it's the same muscle groups that are involved in both a voluntary movement and emotional movement. It's not like you have separate muscle groups that do emotional smile versus voluntary smile. They react in slightly different ways, which is why we can see the differences. But it's the same motor pools. The difference in the overall activity is dependent upon the fact that they originate, the signal originates from different sites, right? So the voluntary movement coming from the motor cortex, and then we have a separate set of areas coming from extrapyramidal productions from the medial forebrain and the hypothalamus. So a lot of the emotional areas of the brain that regulate the emotional smile. And when there's damage on this circuit, this leads to emotional facial paresis, like in that boy. And this is referred to as a Duchene smile because of a scientist who had studied this phenomenon. His last name is Duchene. Both are going to affect the motor pool, but they're going to affect the motor pool in different ways, and especially depending upon whether someone's trying to do a voluntary smile or is doing a spontaneous smile. So it demonstrates that there's selective lesions in one of the two anatomically and functionally distinct sets of descending projections that motivate the muscles of facial expression. So there are distinct reactions from the autonomic nervous system, the sympathetic nervous system in particular that we're talking about here, in reaction to minor stress versus intense stress. So stress is not an all or none category. There are degrees to which one experiences stress. Minor stress, so here's the arrow showing when the minor stress occurred. We're going to see some effect on physiology. You're going to see some subtle changes in heart rate, that it's relatively elevated compared to the baseline, just a bit. There's an increase in perspiration initially, and the respiration seems to increase a little bit too. Under more stressful stimuli, we see a substantial increase in perspiration. We see a substantial increase in heart rate. And we see a quickening of the respiration response. This is showing the degree to which we can have a stressful response. This is going to be tied in to the degree to which one experiences just simple discomfort versus a more salient experience that will be related to anger, fear, or other things. And so this is that underlying physiological change. So again, the components of an emotional experience is this subjective component, where we can ask the question, what is the person's internal emotional experience or response to a stimulus? In this case, this person is experiencing fear and horror. The physiological response. What are the physical symptoms, as we just discussed? Increased heart rate, sweating palms, rapid breathing, presumably what the scream guy is doing. And then behavioral and expressive response. What does the person actually do? Facial expressions are indicative of fear and distress and screaming, hence it's called the scream. So again, these three components are key to the emotional experience. There's some early theories of emotion coming from the cognitive psychology parts of addressing these issues. And they debated the relationship between emotions themselves, say as the subjective feelings of fear, and related physiological responses. There's the James Lange theory, which suggested that emotion was experienced in response to physiological changes within the body. So in this theory, the idea is that there's a sensory stimulus. Seeing a snake, the stimulus is perceived. And then within the James Lange theory, there is this, the emotion is in response to the physiological changes. So because the stimulus perceived will induce a visceral response, we generally have a natural inclination to be fearful of snakes. It's going to induce a physiological change within the body. We perceive that change in our underlying physiology, and that perception of the physiological change is what shapes our emotional response. That we will say we are feeling fear, and that can also have a control over the behavioral output. The Cannon-Bard theory argued that emotions occur independent of the emotional expression, that there's no correlation with the physiological state. So the idea here is that we perceive a stimulus, and that's going to lead to those emotional characterizations within the brain, and it's not dependent upon having that first physiological response, and that in fact the physiological response is dependent upon that the concept of fear first. So I've set the stage here for the cognitive understanding of emotion and fear and emotional perception, emotional response. There's obviously a complex set of brain areas that are involved with this process. Our best understanding of the underlying systems that regulate emotional experience and the visceral and autonomic responses that are tied to the emotional experience is known as the limbic system. So we're not going to go through all the details of the limbic system, but I do want to walk through the underlying components to this. So the limbic system is generally referred to these structures that create this kind of crescent shape on the medial side of the brain. And it involves a whole bunch of substructures. So limbic is the Latin word for border or rim, and that's because this is the areas that are a ring around the corpus callosum. So the corpus callosum is here. This is the bundle of axons that connects the left hemisphere to the right hemisphere. Right? We've discussed this in great detail with split brain patients and how that when this is split, that we can understand some of the differences in how language is perceived from one side of the brain to the other side of the brain and how it's regulated. So, but the limbic system is the area around that. And so it's this crescent around it. And so the border, the limbus. And it involves a bunch of different areas. So we've got orbital and prefrontal medial cortex. Orbital and medial prefrontal cortex on the medial portions. We've already talked about some of the role of the orbital and medial prefrontal cortex when we talked about executive function and decision making. It also is a component of the limbic system. We talked about the cingulate gyrus. The cingulate cortex, the anterior cingulate cortex, and the posterior cingulate cortex being components of executive function. They also feed into the emotional experience, and they are considered part of the limbic system. We have portions of the hippocampus, the parahippocampal gyrus, portions of the temporal lobe. The amygdala is a key component to this. This is the amygdala here. And then also subdivisions within the hypothalamus are also part of this as well. And the thalamus. So we're going to touch on some of these areas, and then eventually we're going to be discussing the amygdala in detail. So much of this work was identified by the neurologist James Pepys. And so this set of, and he did a ton of this early work, and so these limbic regions are sometimes referred to as the Pepys circuit. So again, a quick little summary of the thalamus. This is a figure we've seen a few times, right? We've talked about the thalamus being numerous subnuclei that are grouped based on function, that we have sensory relay nuclei in blue. We got the motor relay nuclei in red. So we talked about that in the first two parts of this course. Now we're going to talk about the limbic nuclei. So there are subdivisions within the thalamus that are connected to the limbic brain regions. And so some of these nuclei are known as the dorsal medial nucleus. So it's on the medial portion of the thalamus, and the anterior nucleus. And they participate in emotionally relevant information. That's about as much as we're going to talk about on the thalamus. But just so you know, right, that we have relay, just like most other areas of the cortex that involve sensory perception, motor output, that we also have the emotion is an important part of what our bodies and brains do. And it ties into having to relay information to the cortex. And of course, that's going to have to be passed through the thalamus. And so the thalamus has major subdivisions that are dedicated to contributing to regulation of emotion. And those are the limbic nuclei of the thalamus. So let's get a little closer looking at some of these limbic structures. So we have these thalamic nuclei that are really important for emotion, as we talked about. The anterior nucleus of the thalamus, medial dorsal nucleus of the thalamus. We have the ventral basal ganglia that are related to motivating behaviors. They are going to feed into our overall emotional state. We talked about these: the ventral tegmental area, the VTA, and obviously the nucleus accumbens. They obviously are going to play some role in our overall emotional state, and particularly in rewarding um, uh, the positive affective type of stimuli, giving us a positive, happy mood related to positive stimuli like food and sex. The hypothalamus features into this as well. We talked about this when we talked about stress, that it regulates the endocrine and autonomic responses to certain emotional stimuli, and will have an influence over emotional behaviors such as aggression, say via vasopressin. The amygdala, which is what we're going to discuss in great detail in a few slides here. It processes emotional behavior responses such as fear, not only fear though. It drives the formation of fear memory and coordinates autonomic responses to fearful stimuli. It plays a very important role in fear, but more data has shown that it also regulates other aspects of emotion as well. The mammillary bodies we have mentioned before, they play a role in memory formation related to emotion. And then the hippocampus. It governs the learning and memory of placing emotions within a context of a situation. Remember, the hippocampus is really critical for tying in the episodic components of memory. And the hippocampus, obviously is going to help give us that episodic components related to the emotional context of a situation. And it can shape stress responses and also have an effect on anxiety-like behaviors. Other aspects to it is the fornix, which is a fiber tract connecting the hippocampus to the hypothalamus, septum, and ventral striatum. So here's the fornix here. So this is a fiber tract connecting the hippocampus to those structures. And then the mammillothalamic tract connects the mammillary bodies to the anterior thalamus. So I'm not going to ask for great details on most of those structures. You should just be familiar with the list. Instead, we're going to focus a lot on the amygdala for the rest of today's presentation. So a little bit about the amygdala. It's critical for processing most emotional stimuli and guiding emotionally relevant behavior. It's situated at the pole of the temporal lobe. So it's within the temporal lobe, and it sits at the tip of the hippocampus. Remember, in the human hippocampus, it's on the more ventral side of the brain along the temporal lobe, and the amygdala sits here. And it has kind of this almond-like shape, and in fact, that's where it gets its name. Amygdala is the Greek word for almond. Good name for it. I think amygdala is a nice name. It's subdivided into several subnuclei. There are 3 broad regions within the amygdala. So if you look at a cross-section in the coronal plane, you can see these 3 subdivisions, depending upon where you are on the posterior to anterior axis. But here's a nice big cross-section here. There's a bunch of different subregions. The amygdala is actually quite a complex structure, but we can break it into 3 major subdivisions. First is the cortical medial nuclei, excuse me. It communicates with the olfactory cortex. So this is the cortical medial nuclei located here on the more dorsal side of the amygdala. A big chunk of the amygdala is referred to as the basal lateral nuclei. It receives visual, auditory, gustatory, and tactile information, and it also connects to the central nucleus. So here we have 2 of these major areas that just happen to receive input from these different sensory modalities, right? So the amygdala, it has to perceive the world, and it's getting information from all of our sensory modalities. It just happens to segregate the olfactory information from the other information. The reason why it's segregated, if you think about this, it's because these receive inputs directly from mitral cells and not through the thalamus. Whereas the rest of this has to be gated through the thalamus, and so it's coming from likely other areas, maybe even cortical areas as well. So that's part of the reason why there's this major division within the amygdala. Then the central nucleus. It's connected to the hypothalamus and the brainstem, and it regulates the coordination of the autonomic output. So in general, information comes in through these two major subdivisions. There is a ton of processing that happens within these two areas. And then in general, the output of the amygdala is going to be the central nucleus, where it's going to have an effect on hypothalamus and brainstem output, which is going to have a major influence on our emotional response. So the information comes in and is processed through the amygdala, and then it comes out the central nucleus. So just like basically every brain area that we've talked about, one way that we have learned about the amygdala is through the process of just lesioning it. So we can look at the effects of amygdala lesions. So a bilateral amygdalectomy reduces fear and aggression in all animals tested. This is in non-human primates, rodents, cats, and other species. So this seems to be a pretty consistent phenomenon that the amygdala is critical for overall fear and aggression response. But we've seen a really unique case of a very specific kind of amygdala lesion, an amygdalectomy, so to speak, in a very famous neuroscience case. This is one that maybe you haven't really heard of, unlike patient HM and Phineas Gage. She's a little less famous, but she's still a very important case. So this is a human subject called SM. She suffered from a rare condition called Urbach-Wiethe disease. So in this disease, it causes calcification within the brain. So you get a little bit of bone growing within the brain, and it can cause bilateral calcification and atrophy of the anterior medial temporal lobes. So this particular individual happened to have calcification that caused extensive damage to her amygdala. So the calcification sort of just cut off circulation to the amygdala, so they no longer got any blood flow, and then the amygdala just died. And surprisingly, in her case, it was really kind of just specific just to the amygdala, and there really wasn't much damage to the other nearby areas, including the hippocampus and the cortex. So really this was like a perfect amygdalectomy. You couldn't even really do this through like surgery very well without damaging nearby structures. So she was kind of a perfect test case. She had no motor or sensory impairment and notable deficits in intelligence, memory, or language. Her deficiencies were strictly within the emotional realm, specifically the emotion of fear for her. She doesn't seem to recognize or to experience fear, nor can she recognize it in others. So if you showed her fearful faces, she doesn't recognize the emotion that they show. So the overall perception of seeing fear in other people apparently is also dependent upon the amygdala. Some of the experiments asked whether SM could experience fear in real life situations. So kind of like patient HM, she was put through all kinds of tests, and she was exposed to a number of situations that typically evoke fear responses. She was taken to an exotic pet store and asked to handle poisonous snakes and spiders, and then they just monitored her behavior. She was just intensely interested in them, and she showed no signs of avoidance. This is snakes and spiders is just something that people will instinctually be afraid of. Of course, we also train through experience kids to be afraid of snakes and spiders. And so, this can lead to enhancing those natural instincts. You can also fight against them too, obviously. With experience, you can learn that, like, okay, handling poisonous snakes and spiders can be done carefully. In general, most people are going to have fear. She had no fear whatsoever, despite the fact that she never really had much experience with poisonous snakes and spiders. In a haunted house. They took her to a haunted house. So this is another test. I want to study SM. Get to go to the pet store, get to go to the haunted house. This sounds like good research. All right, so they took her to a haunted house. You can just imagine like a bunch of nerdy dorks with clipboards following in white lab coats, following SM around in a pet store and haunted house. So they took her to a haunted house. She led people through the house, showing no signs of nervousness or fear. She wanted to approach and touch the monsters. Pretty funny reaction. Overall, these experiences did not evoke any fear, but rather piqued curiosity and aroused excitement. So she had kind of the arousal aspect to it, but her perception was not of the experience was not negative. Instead, it was a positive experience. Sort of, she described it as like feeling like when riding a roller coaster, for instance. So it felt like that. So nothing scary about it. It was just kind of fun. In another study, they had SM perform a daily emotional assessment over a 3-month period to rate her emotional state. So at random times throughout the day, she was asked to just write down what kind of emotional experience that she was feeling at that moment. And the results showed that she had a rich emotional life that reflected most human nuances of emotion and mood, except that she didn't ever really experience fear or being scared at all. So you can see the major emotions: sad, angry, surprised, happy, disgusted, afraid, that are cross-cutting. And then when you divide them into some of the subdivisions that we associate with these major divisions, such as sad, loneliness, and downhearted, angry, irritable, and upset, she was able to experience some of these things. Feeling surprised, happy, excited, and cheerful, and even disgusted. She could have that as well. But one thing that she couldn't experience was fear. So clearly her ability to feel afraid was quite compromised. Another study had SM draw facial expressions of emotion. Sketches at the right suggest that SM could conceptualize emotions as expressed through facial expressions, but she had no way to conceptualize fear and instead drew the body and not the face. So our face, there's a universal way of expressing fear through our face. Raising our eyebrows, opening up our eyes, having big open eyeballs so that you can see the whites, tensed lower eyelids, so bringing those eyes as wide as possible, and dropping our jaw slightly to kind of make almost like a scowl like look or an unhappy face. So she was asked to express happy, sad, surprised, disgusted, angry. She could do these pretty well, but she couldn't show afraid. It almost kind of looks like an angry baby crawling. Definitely not. She couldn't draw the face of being afraid at all. Even though the surprised shares a lot of similar features to being afraid, she couldn't do it. So lack of fear, including even perception of fear. So, of course, when we talk about emotion, we're talking about things that are obviously very relevant to the human experience, as opposed to when we talk about other behaviors that definitely humans have and show, and we can kind of find easy correlates in animal models. When we talk about rodent animal models for emotions, it becomes a little harder. I mean, this is what a rat looks like when he's happy or sad, excited, disgusted. Point is, how do you know? You can't really tell a whole lot about the overall emotional state of a rat by just looking at its overall facial expression. That said, there are certain ways that we can assess other kinds of behaviors that will give us some insight to emotional state that does have distinct correlates to humans. So we're going to talk about some of the kind of experiments that are done in rodent models that we know are the correlates to those things in humans. So there are ways of testing anxiety in experimental animals. This is essentially fear test. So there are two main tests for doing this. One common test is known as the elevated plus maze. This is here. This is the elevated plus maze, so it's up quite high. And this is a half a meter up, and it's a plus maze. So there's just like basically two paths that the rat can take. There's and one of the paths, one cross section of the plus is enclosed. So it's entirely closed up so the rat doesn't have to look over the side. And the other part is open, where the rat could look over the side and see how high up it is and kind of be scared. The open field test is one where it's just a big box, rat can't get out, and you look to see where the rat is within the open field. Is the rat going towards the middle or going to the outside? So basically, both of them are testing anxiety related to space. In general, rodents do not like to be out in the open. Being out in the open means that they're more likely to be seen by a predator, such as like a hawk or an owl, and that the predator could come in and swoop down and just snatch them up and eat them. So they like to be enclosed. So you're going to find that rats, especially more anxious rats, are going to spend more time in the closed parts of the plus maze than in the open parts. And they're not going to explore a whole lot. Some rats, though, are going to be a little bit more adventurous and a little less anxious, and they will spend more time exploring the open arms. And so you can see them spending time out here and just looking around and not being so scared. They'll go into the closed parts too. And then you can compare those things to see the degree to which they're anxious. So if they have a little exploration overall, that indicates high anxiety. Exploring the risky areas indicates limited exploration of the risky areas indicates high anxiety. And if they take a long time to explore the risky areas, that also indicates anxiety. If they're hiding in here before they go out here, they're anxious. The same can be true for the open field. So in an open field test, a rat or a mouse is going to spend a lot of time in the corners and a lot of time on the edges and not spend a whole lot of time in the middle. So a particularly anxious mouse or rat is going to just basically stick to the outside and not go in the middle. A rat or a mouse that is a lot less anxious is going to spend time in the middle. You might even put, say, a novel object, which rats and mice tend to want to inspect. You can put it in the middle and you can measure how quickly it takes them to approach that novel object. Anxious rats and mice, they'll ignore the novel object. They're just going to hang out in the corner, whereas a bold and less anxious mouse or rat will go right up to that novel object and spend some time with it. So these are the different kinds of tests that can be done with mice and rats to test for anxiety and fear. So this is what this looks like. This is actually some images from the Clinton lab. So Dr. Sarah Clinton is a professor in our School of Neuroscience. She studies, one of the things that she studies is that she has these two lines of rats that have been bred to either be less fearful or more fearful. So she's got low responder rats that are more fearful and they are less interested in novel objects, such as like a little watering can that they put in the middle here. And then high responder rats, which are much more bold and a lot less anxious and will cross into the middle and spend their time in the middle. So this red line here is just showing the path that the rat takes over like a 5-minute period. And so you got a camera on top of the ceiling, and you can just monitor how much time the rat spends in the middle versus the outside, and also like how much movement that they're doing or whether they're just sitting in the same place. And what you can see is that, so if you're just looking at, say, time spent in the center to inspect a novel object, the high responding rats are more likely to spend time in the center. They spend around 15% of their time in the center versus the low responding rats, which is around 5% of the time. This is a significant difference. So she's got these two lines of rats that show this significant difference in the degree to which they are anxious. The other interesting thing is that if you treat them with a chemical that's known to decrease anxiety, so an anxiolytic is what it's called. It reduces anxiety, called benzodiazepine. If you give this to both sets of rats, it doesn't really have a whole lot of effect on the high responding rat because this rat is already kind of like not so anxious. But the low responding rat, if treated with this anxiolytic drug, now spends time in the center, just like the high responding rat. So these anti-anxiety drugs can that are known to affect anxiety in humans. They can also affect anxiety in animal models. So in the elevated plus maze, this is showing some of these data from Dr. Clinton's lab, just looking at the high responding rat versus the low responding rat. So this is showing the open arms here. So these are the open arms, and then these are the closed arms. And this is just showing the path that these rats take. You can see that in the for the high responding rats, these guys are not anxious. They spend a lot more time in the open arms. They're up there looking around, seeing what's up, what's going on the outside. Whereas the low responding rats that are super anxious, they spend almost all of their time in the closed arms. You can see they kind of just peeked out a little bit to see the open arms, but they didn't even bother to go in the open arms. So again, these rats that, and there are all kinds of other correlates that go with being a low responding rat versus a high responding rat. These guys tend to be a lot more bold and also a little bit more easy to show addictive like behaviors versus these guys. So there's some really interesting behavioral differences between these two kinds of rats, but a lot of it just comes down to overall anxiety. So this is a complex circuit showing how information passes through the amygdala. So I had a sort of a brief description of what this looks like. So I just wanted to show this to give you a sense of how information is passing through the brain to get to the amygdala. Like I said, the amygdala receives sensory input. It does this through the basal lateral nuclei and the cortical medial nuclei. And in this diagram, it's not even really showing the cortical medial nuclei. It's just showing the basal lateral nuclei, since that's where a ton of information is going through this. So for instance, auditory information can pass through, if we're just talking about auditory information, what you're going to find is that you're going to have, so like a scary noise, a ghost making ghost noises, or a zombie making like brains. That's pretty scary. Goes through its normal auditory pathways. It goes through the auditory thalamus, and there's inputs directly into the basolateral nuclei. There are also, of course, other inputs from cortex into the basolateral nuclei as well through other cortical areas. So not only do you have the direct thalamic input, say like you hear a gunshot, you're going to have an immediate response that will pass through the amygdala that will give that fear response. It goes through the amygdala to do this. And then there's some processing on what does that mean? Oh, geez, that noise I heard that was really loud, that was scary. I recognize that as a gunshot. That's not good. I'm actually pretty scared. It's going to go through your cortical areas and be recognized as a gunshot, processed through certain association areas that will lead to the also informing the basolateral nuclei of the amygdala that say, you heard a gunshot, this is bad, you really need to have a fear response and get the heck out of here. So the basolateral nuclei, there's a whole bunch of subdivisions, will process this information, and then that information will be sent to the central nucleus, which is then the output. So then from there, it can feed into the endocrine response to lead to the HPA axis. It can also inform the HPG axis to have an increase in testosterone in men to allow them to fight. So some of this is driven through the central nucleus as well. An autonomic response, initiating that quick sympathetic nervous system response that gets your heart rate going immediately. That is done through this connection. And then a behavioral response. So the central nucleus will have automatic control over certain behavioral systems that'll give an automatic response so that you can have a quick response to a sudden stimulus. So just to summarize that, the central nucleus is connected to the hypothalamus and brainstem to coordinate autonomic output, endocrine control, particularly the stress response, but it can lead to changes in testosterone as well, and behavioral output to shape emotional responses. One key component to emotional events, in particular those in which elicit fear and are particularly stressful, they can be established as long-lasting memories. And these memories are, they can be particularly vivid and very long-lasting. So this is almost like, I mentioned this on the exam, a flashbulb memory. When you are confronted with a particularly stressful event, this is going to include activation and of the amygdala. The amygdala, it's one of these critical brain structures that's important for the establishment of emotional memories. The hippocampal system is important to this as well. So all of the episodic and explicit memories about the emotional situation will be tightly laid into the hippocampus. But the overall fear and the reaction and shaping how one is confronted with similar stimuli in the future, this is shaped and laid down in the amygdala. This is referred to as learned fear. So let's talk a little bit about how that works. It can use a very simple sort of conditioning paradigm. And we'll walk through precisely how this works. So this is a very classical condition. This is using the principles of classical conditioning that we've already learned about, but in the context of fear. So a fear response in a rat is going to include an increase in blood pressure and an increase in freezing behavior. So a rat that is scared is going to freeze. He's not going to move around because a rat that's moving around will be easily seen by a potential predator and be snatched up and eaten. So freezing allows them to blend in with the background a little bit, and they're just going to not be seen as likely. So that is the typical rat fear response. So one way of testing this initially, it starts with getting a baseline. So you play a tone. So like you have a little speaker in here, it plays a tone, the rat hears a tone, and there's some change in blood pressure because like, oh, that's weird, hearing a tone. And it's going to freeze a little bit, but not a whole lot. So about 1 second out of every 10 seconds. So there's little to no overall response to the tone. Then we do the pairing. So in this case, the rat hears the tone and receives a foot shock at the same time. So this rat is in a cage that has an electric grid below, and this is going to induce a strong fear response. It's because the foot shock is very scary. So blood pressure shoots up, and now the rat is freezing 8 out of every 10 seconds. So very scared. After doing this, you can really only have to do it once. But if you do it a couple of times, it's even more salient. The tone itself will elicit a negative response. So just by playing the tone, you're going to have an increase in blood pressure and an increase in freezing relative to when the rat heard the tone before. So this is post-conditioning, and it's now induced a strong fear response to the tone. So let's walk through this in a little more simplified fashion, but taking into context what the amygdala is doing. It is correlating the electric shock plus the tone, connecting these two things together. And some of this connection is laid down within the amygdala, which is going to shape the fear response. So it takes in this sensory information and processes it, connects it to lead to activation of the hypothalamus to induce an autonomic response through the sympathetic nervous system. It's also going to lead to brainstem areas to induce a behavioral reaction. And it will also impact the cerebral cortex as well. There's some impact through other aspects of the amygdala to shape the emotional experience. There have been some really cool experiments in neuroscience. I mean, just overall, some new techniques have really shaped the way that we understand neuroscience. And we've talked a little bit about optogenetics using calcium imaging, where you can look at the overall activity of neurons by looking at changes in fluorescent signals related to calcium. We've looked at a few experiments like that. So we're going to talk about another form of optogenetics, which is going to be used to regulate neuron activity. So this kind of optogenetics uses a combination of techniques from optics and genetics to control the activities of individual neurons in living animals. It uses what's called channel rhodopsin. So this is a channel that is taken from rhodopsins. And I've talked about this before, but I'm just going to go through it again because this is a very important critical technique that's now being extensively used in neuroscience. It is sensitive to light. In this case, this particular channel rhodopsin is sensitive to 470 nanometers of light. When you shine light on a neuron that it's expressing channel rhodopsin in the cell membrane, so this neuron has channel rhodopsin all over it, you shine light on it, it's going to open up, it's going to allow sodium to flow in, which is going to induce, as long as you reach threshold, you'll induce action potentials. So you can depolarize the cell and cause action potentials. So by a simple way to do this is to implant a fiber optic cable into the brain, which you can shine in the light, and it'll induce activity in particular brain areas. So this has been done in many different brain areas. It's revealed an awful lot about how nervous systems work. It's also been done in the amygdala. So we're going to talk about one really cool experiment where they turn on and off the light to change activity within the amygdala to shape overall activity. So in this experiment, they're actually they're able to activate certain parts of the amygdala. So now they're getting down into specific areas of the amygdala. They have 3 different kinds of animals that they did this in. So they have one group of animals where they were put in channel rhodopsin into neurons that connect from the basolateral amygdala to the central amygdala. So there's a subgroup of neurons that connects from the basolateral amygdala to the central amygdala. Another group, another experimental group that they did, they're shining the light on just the neurons that would be leaving the basolateral amygdala into the central amygdala. But these neurons are expressing not channel rhodopsin, they're just expressing a fluorescent molecule called yellow fluorescent protein. So this is to control for the fact that these neurons have been transfected. They use a lentivirus to get in there, and that's going to change some things in the cell. So you got to control for that. So instead of controlling, transfecting these cells with channel rhodopsin, which is the CHR2, they're just transfecting them with a yellow fluorescent protein. And so it's not going to change the activity of the cells, but it controls for all of the surgery and for the cells being infected. Then they have another group where they transfected the cells in the basolateral amygdala. And so the basolateral amygdala, remember, it's made up of all kinds of little subdivisions. And so there's a bunch of different types of neurons that are transfected in here. And then if you put the light directly in the basolateral amygdala, you'll be activating just those neurons, including the neurons that would go to the central amygdala, but all the other neurons as well, not just a subgroup. Doing this activates a subgroup. This activates a big, vast swath of neurons. Okay, so this is more precise than this. Okay, so in this experiment, they had mice that were anxious. They spent a lot more time in the closed arms than in the open arms, and they did this for 5 minutes. And during this time, the light is off. Then they turn on the light in the 3 different groups. They start shining the light. And what you can see is that some of these animals, all of a sudden when the light is on, is now less anxious. They're able to control their anxiety by now they're spending more time in the open arms when the light is on. And then when the light is back off, then the mouse is now back to being anxious again and spends all its time in the closed arms. So the 5 minutes off, 5 minutes on, 5 minutes off. And they do this in the 3 different groups. What they found is that if you activate the neurons coming out of the basolateral amygdala into the central amygdala, if you turn them on and you have them stimulating and they're firing more than they normally would, this increases their probability of going into the open arms. So this subunit of neurons that is the output of the basolateral amygdala to the central amygdala, this is driving these animals to be less anxious. If you do it in the basolateral amygdala itself, you don't see any of this significant change because this is including a whole bunch of different other groups. And so it's not changing the output in any sort of regular way. But if you're changing the output of the basolateral amygdala to the central amygdala, you can induce a decrease in the anxiety of these animals. Let's look at what happens when we do the same kind of thing, but in the open field maze. So we have the same 3 groups with the light stimulating neurons coming from the BLA to the central amygdala. the control group, and then stimulating neurons in the BLA itself, but not that subunit of neurons. So here's an example of one mouse when the light is turned off in this group. You can see that this mouse is staying to the outside, not showing a typical fear response. When the light is on, now the mouse is showing less of a fear response. And when you turn it off and on and off and on and off and on, you can turn it off and on like clockwork. You can see that when the light is on, the group with that subunit of going from BLA to central amygdala is showing more time in the center. And then when you turn it off, it goes back down to the controls. On, more time in the center, off and on. Now I realize this is somewhat counterintuitive, but what it means is that an increase in the neurons, an increase in the activity of the neurons from the BLA to the central amygdala is associated with decreased fear response. And what this means is that the fear response is dependent upon, at least in part, a decrease in activity on the central amygdala from the basal lateral amygdala. I realize this is kind of somewhat counterintuitive because we normally think of, oh, well, activity has to be related to inducing this behavior. But instead, what seems to be the case is that this is one regulation point where, in fact, it's a decrease in activity that leads to this. And so this is also correlated to some of the changes that we see in amygdala over stress. That's shown here. So in this experiment, what they did is they exposed mice to 21 days of chronic stress. And this led to a change in the dendrite length in the amygdala. So, but the kind of change they saw in dendrite length depended upon what part of the amygdala that they were looking at. So there's a portion of the amygdala called the medial amygdala. And in that portion, chronic stress led to a decrease in dendrite length. This is an indication that these neurons are sort of being suppressed. And then for the basal lateral amygdala, chronic stress led to an increase in those neurons and their overall dendrite length, which indicates that there's more activity and that these neurons are being sort of hyper-excited under chronic stress conditions. So it really is consistent with the idea that the amygdala is a very complex structure that regulates emotion and like fear in complex ways, and is not just a simple fear area where you're just going to have this whole thing, increased activity, more fear. It doesn't work that way. It's actually really complex on the inside of the amygdala. And that's what the previous experiment shows, with the increased activity in the amygdala being associated with less fear and with greater resilience to fear. And in this case, seeing very different kinds of responses in the structures of neurons getting smaller or larger, depending upon which part of the amygdala you're looking at, in the same animals that are getting chronic stress. So the amygdala is amazing, and it's complex. So there's a number of key questions about emotions in the amygdala. What are the three components of emotion? What distinguishes a genuine smile from a voluntary smile? And what are the effects of disorders on parts of circuits related to smiling? What are the major areas of the limbic system? What are the three areas of the amygdala? What is the effect of an amygdala lesion in humans? You should know the details of all of that. What are the key behavioral experiments used in rodents to test for fear and anxiety? What is the effect of anxiety of changing activity of neurons in the amygdala? So that's the channel rhodopsin experiment. And what is the effect of chronic stress on dendrites in the amygdala? So that's it. We'll see you next time.